Alright everyone, welcome back. This chapter we are going to be covering an overview of the nervous system and nervous tissues. Here are some of the learning outcomes. These are our goals and objectives. By the end of this discussion, this lecture, you should be able to be familiar with all of these. And here are a few more on this page. So our first section is just going to be talking about the basic structure and function of the nervous system. We're going to classify the nervous system in different ways, and then we'll dive into the different cells of the nervous system, and then we'll talk more about physiology. The nervous system is designed to monitor everything that's going on inside the body, but also outside the body. If you're driving a car, you need to be able to understand how fast you're going. You need to be able to move your eyes multiple times in multiple areas. You need to be looking at the road. You need to be checking your mirrors. You need to be checking your speedometer. You need to be always constantly making adjustments to make sure that you're not veering off the side of the road or in somebody's lane. And that's what your brain is constantly doing along with the other parts of your nervous system. So what actually makes up your nervous system? As I just mentioned, your brain is kind of the main thing that we think of that's in charge of the nervous system. Your spinal cord, and what I'm going to do is I'm going to add this structure between the brain and spinal cord. We're going to differentiate this next chapter specifically, but the structure right between the brain proper and the spinal cord is called the brain stem, and I'll just put BS right now for brain stem, but that's going to be very, very important. You can technically live without a brain. You can technically live without a spinal cord. You can't live without a brain stem. And then we have nerves, and nerves are just all of these things attached to the brainstem and spinal cord, and you can see them in purple. They're all just branching off, and they're going down to your arms and your legs and your torso region, even in your head, face, and neck area. So we'll talk about all of these different structures within the next 2 chapters. We can compartmentalize the nervous system in many different ways. One of them is just anatomically. Just like we did with the bones. Remember, all of the bones pretty much in that mid-sagittal line we said was the axial skeleton, and then everything else to the side for the most part was the appendicular skeleton. It's kind of the same thing in the nervous system. We have, from a midline or a central portion, we have something called the central nervous system. So this consists of your brain in the middle of your head, your brain stem that connects your brain to your spinal cord, and then I'll do the best I can to draw a straight line all the way down the spinal cord, although with a mouse it's pretty difficult. There we go. And the spinal cord actually technically ends right there, but there is tissue that does continue down all the way to like the coccyx, the tailbone. But basically everything in red that I just highlighted, that's the central nervous system. And as you can see, it's compared to the rest of the body, the arms, legs, and torso, it's actually kind of a very small amount. But what's going to be really interesting is most of our neurons are actually going to be found here in this central nervous system. So this is the structural and functional center of the nervous system, meaning this is where we do all of our calculations, our movement. This is how you perceive touch and pain. This is how you hear things and smell things and taste things. Without your central nervous system, specifically brainstem, and your spinal cord to connect things, pretty much all of it, you can't really live a quality life. You could, like I mentioned, you can live without certain parts and be in a vegetative state or a coma, but to live and do daily activities, you need all parts of the central nervous system working together. And then everything that is not brain, brainstem, or spinal cord can be considered part of the periphery, or what we call the PNS, the peripheral nervous system. And the peripheral nervous system just innervates or goes out and touches everything else in your head, face, neck, arms, legs, torso, even your organs. So We have, as you can see in the notes, two different origins. These nerves is what we call them. These nerves can branch either from the brain and brainstem area. So let me add that, brain and brainstem. And if they do that, they're called a cranial nerve. And these are the nerves that generally go to your eyes, ears, nose, throat region. We'll talk about all 12 cranial nerves eventually. But examples of this is like your optic nerve that helps you see, your facial nerve that helps you smile, that kind of thing. We'll talk about the rest. Everything that is branching from the spinal cord. So in green, you can see all of these many branches going down the arms, branches along the rib cage, branches down the legs. Those are all considered spinal nerves because they're branching directly from the spinal cord. So while we are separating these two nervous systems, we have CNS and PNS. Please know that they're all connected. In order for you to move your toe, your brain has to be connected to your toe. So as a message, what we're going to call a nerve impulse or an action potential, as the message goes down your brain, it has to go down your brainstem, down your spinal cord, and then out down the nerves to your leg all the way to your toe. It's connected. And then when you cut your toe or when you stub your toe, there's also nerves that just go the opposite route right next door, and they just go to a different part of the brain. So everything's connected. CNS and PNS are intertwined as one nervous system. It's just from an anatomical standpoint and from a functional standpoint, we can separate the two. Here are some functions of the nervous system, and as you see, communication, control, integration. You can make the argument that all three of these words are intertwined. They can all kind of point towards the same thing. Basically, it just means that the nervous system oversees all operations. It's the CEO or the boss of your entire body. But to give you specific examples, communication is just how we talk from the brain to other cells. So once again, I can move my toe. I can move my mouth right now because my brain is telling me how to move my mouth. I can blink. I can scratch my neck. Whatever I can do, there has to be messages or electrical impulses that actually go to certain parts of the body. And we learn how this works with the muscular system, right? If I want to contract a skeletal muscle, it all starts in the brain. I have to send an electrical impulse to that muscle, and then we can release the acetylcholine, and then the sodium can go down the T-tubules, and the sarcoplasmic reticulum can release calcium, and all those things we've already discussed. But it all starts with the brain communicating with the muscle. The brain and the nervous system also controls things. We talked about homeostatic mechanisms in Chapter 1. We talked about the difference between sweating and shivering. Regardless if you're sweating or shivering, your body's trying to get us back to a normal body temperature, that 98.6, give or take a degree. We talked about blood calcium levels recently. If your calcium's too high inside your blood, that's really, really bad. Hopefully we're going to store it in the bones. The brain is what dictates all of that. It's what's controlling that thyroid gland and those parathyroid glands. If your blood calcium's too low, we need to fix that. And the brain's going to tell the body to release some of that calcium, right, from the bone into the blood. So controlling the food that you ate this morning for breakfast, your body, your brain, your central nervous system is controlling how you digest that food, how quickly and how you absorb those nutrients. And then last but not least, integration. I kind of already just discussed this. It basically just everything works together, right? If we are working out, say that we're really pushing ourselves, we need multiple organ systems to work together. We need to, one, get more oxygen to our cells, specifically our muscle cells. So one way to do that is make the heart beat faster. Another way to do that is breathe faster and breathe deeper, right? Get more oxygen in your body. Another great way of getting the oxygen and nutrients to your cells faster is to increase blood pressure. And we can do that by increasing heart rate, but we can also do that with the kidneys, with the urinary system, as we'll find out. The urinary system is going to be told to hold on to fluid, and the more fluid you have in your body, the higher the pressure. So we just tied in nervous system with cardiovascular, with respiratory, with urinary. All of these things work together to simply help you run down the road for a workout. And there's many other your bones and muscles and every other organ system is working to do that too. So basically, recapping this slide, your nervous system is running the show. They are in charge. They are the boss. Everything else in your body is just kind of a puppet, if you will. So on this slide, I want to cover how we can organize the nervous system from a functional standpoint. And like I said, there's many different ways we can do this. But what I want to do is I want to first talk about the difference between sensory and motor. So I'm going to put a sensory division over here. And then a motor division on the right. And pretty much every single nerve impulse, except what's going on between the brain itself, different parts of the brain, but anything that the brain is talking to, such as the liver or the pancreas or the kidneys or your muscles, that's considered a motor response or a motor message, right? My brain tells my fingers to motor, to move. My brain is telling my heart to beat, to contract. That's a motor message. A sensory message is just anything that you sense. Something's touching your skin, something smells funny, I'm talking really loud in the microphone, you taste salty crackers, or whatever. That's just something coming into the brain that you're sensing, some type of stimulus causing that. So what I'm going to do now is we're just going to put some nervous system terms to these words. Anything that has a sensory message that's coming towards the brain or towards the central nervous system, what we call that is an afferent message. And then anything leaving the brain going out to an organ or muscles count as organs, right? Anything leaving the brain would be considered an efferent message. Now, very similar to the joint movements, we talked about adduction and abduction a couple chapters ago. They sound pretty much alike, they're spelled pretty much alike, so we tend to enunciate the first couple letters. We do the same thing here, at least I do. Instead of saying afferent, efferent, I say afferent and efferent. And that's just to make sure that we are all on the same page here. So afferent means it's arriving to the brain or the central nervous system. So I'll put the CNS here. So afferent or afferent is a sensory message that's coming towards the brain. Something's touching your finger has to send a message to your brain. That message is called an afferent message. And then the opposite with the efferent. E is for exit. It is exiting the brain, it's exiting the central nervous system and going elsewhere. It's going to your muscles, it's going to your joints, it's going to your stomach, whatever we're talking about. So you're going to hear me say motor and sensory a lot. You're also going to hear me say afferent, efferent. These words are pretty much interchangeable when it comes to nervous system vocabulary. Okay, so then the next way that we can classify the nervous system from a functional standpoint is whether the nerve fibers tend to innervate or talk to voluntary tissues or involuntary tissues. So we know that our skeletal muscle is voluntary, right? Voluntarily controlled. What we call those types of tissues, skeletal muscle, joints, and the structures around it, we call that somatic. Translates as voluntary. So we have somatic sensory message. We also have somatic motor messages. So that's once again referring to voluntary. Next, we have the involuntary stuff. The organs, right? The heart, the lungs, the gallbladder, the spleen, the the uterus, whatever we're talking about, that we don't really control. And that is referred to as visceral innervation. Visceral meaning organ. So we have messages from the viscera, from your stomach, from your intestines that talk to your brain, and your brain can also talk to your viscera and tell your stomach to work harder or don't work hard. It can tell your heart to increase heart rate or decrease heart rate. So then we can combine these two types of classifications and put it all together and make a big picture of how the nervous system works. So we can send somatic afferent messages to the central nervous system. And this is examples like something is touching your skin. This is something, for another example would be your joint hurts, more of like an arthritis type thing. So skin, muscle, joint, you pull the muscle, that would be a somatic, right? Because your muscles are voluntarily controlled. Skin, not so much, but the skin does fall under this category. So skin, muscle, joints, anything that you feel in that regard is a somatic afferent message. So notice how I put those two words together. Somatic, meaning coming from the muscle, joints, or skin. And because it's sending a message to the central nervous system, to the brain, that is a sensory message, so it's afferent. So we call this a somatic afferent message, or a somatic sensory message. Now we can also do the same thing from a visceral standpoint, right? If you have a tummy ache, your stomach, your tummy is telling the body, the brain, hey, something's wrong here. So we can send a message from the stomach to the brain. Your pancreas can send messages from the pancreas to the brain. Same thing with the liver, same thing with the kidneys, all sorts of different things. Now we do this through both electrical wiring, we can talk to one another through the nervous system, but remember there's also hormones as well. We can release chemicals in the bloodstream, which will eventually get to the brain, which will talk to the brain as well. So we have somatic afferent and we have visceral afferent messages. We can also do the same process from a motor standpoint. We know that we control our muscles voluntarily. So an example of this, a somatic efferent message would be a muscle or a message to your muscle, to your finger. And then as I already mentioned, a message to an organ such as your heart to pick up the pace, or your bladder not to let you go to the bathroom right now, or your pancreas to make more insulin, something like that, would be visceral control, a visceral efferent message or function. So to recap once again, from a structural standpoint, it's real simple. It's just anatomy. We can divide the nervous system up into CNS and PNS. But from a functional standpoint, we have sensory and motor messages. It's basically a big two-lane highway. We got stuff leaving the brain. We got stuff coming into the brain. And then it depends where it's coming from and where it's going. If it's going to the skin, muscle, joint, skeletal system, we can call that the somatic nervous system. If it's going to basically our inner organs, we can call that the visceral nervous system. And this is just another slide or illustration showing you that there are many different ways we can classify the nervous system from an organizational standpoint. You see we have the CNS, brain, spinal cord structures. We also have PNS structures. We'll talk about different terminology and lingo in a chapter. So the difference between a nerve and a ganglion or ganglia, plural. Basically, it's just what part of the neuron are we talking about between those. And then at the very bottom left, you can see that the digestive tract is kind of its own nervous system, just like it's kind of its own immune system. We'll learn more about that next semester. But we have something called the ENS, or the enteric nervous system, where the digestive system can kind of talk to itself solely or independent of the central nervous system. So many different ways of classifying our nervous system. Now, once again, how the nervous system gets their jobs accomplished is twofold. One, it's just simply through electricity, right? Your brain sends an electrical message to something and it gets it done. Boom. The other way we do it is through the endocrine system, through the bloodstream. So remember, endocrine is just referring to some type of chemical or a hormone. It's a chemical floating around in the bloodstream, released from some type of organ or gland. So you do need to know the differences between a nerve impulse and a hormone. A nerve impulse is extremely, extremely fast. I'll give you some numbers later, but depending on what part of the body we're talking about, these can go about 200 to 300 miles per hour. And that's why when you move your finger really quickly, you don't even have to really think about it. It's just boom, move your finger, done, right? That's why you can do so many, when you're running, you can move and contract and relax so many different muscles simultaneously, because these electrical impulses, these messages from the brain, are going at lightning speed. Now, the opposite of that is the slow effect of your hormones, or the slow releasing or processing of your hormones. Now, your blood can circulate through your entire body in a minute, so it's still pretty fast, but relative to how fast the nervous system, where nervous system works in milliseconds, 1/1000th of a second, a hormone could take a couple seconds. So relative to the nervous system, hormones are pretty slow. And because one is extremely fast and one's extremely slow, their lasting effects are different. You can see the nervous system is very short lasting, right? If I tell my finger to move, my finger moves once. I have to tell it to move again and again and again, because the electrical message only does one action. It's just one message and then the message is done. So you have to send multiple messages for the finger or whatever we're moving to move multiple times. Versus a hormone, because it's traveling through the bloodstream, it tends to last a little longer in the body and its effects. Picture adrenaline. Adrenaline can be released, well, it can be released from the nervous system, but most cases it's going to be released into your bloodstream. So if you're driving down the highway or the road or something and somebody cuts you off and you get really mad, some road rage, you freak out a little bit, your blood pressure goes up, you can feel your heart rate, you start sweating a little bit, and then the person sitting next to you says, "Hey, just calm down, it's okay." Mentally, you can calm down within a second or two, right? If they put things in perspective for you, but physiologically, you can't just settle your blood pressure. You just can't stop your heart from beating out of your chest, and you can't stop the sweating instantly. That's coursing through your bloodstream. That adrenaline is moving, and it's going to take a few seconds, a few minutes even, to actually calm down. It's because your blood is constantly circulating throughout your body. It's not a one and done type thing. So in summary here, we have the best of both worlds. We have really fast nerve impulses and really slow hormones. And yes, although the hormones sound bad because they're slow, they last a long time. So if we have some electrolyte issue going on, if we have too much sodium or not enough sodium or potassium, and we drink some fluids or some water or something, your body is also releasing hormones to help control your bladder habits and your thirst centers in your brain, and to basically equalize your water and electrolyte levels. So it's a good thing that this is long lasting. We have the best of both worlds when the nervous system and the endocrine system work together. Let's move on and discuss nervous tissue. Now, a little bit of this should be review. We did cover minor parts of nervous tissue in Chapter 4 with histology, but we will also introduce some new concepts as well. First, let's talk about the neuron. The neuron is the structural and functional unit of the nervous system. So when I say structural unit, that means you put a whole bunch of neurons together, it makes up your nervous tissue. It makes up your brain, makes up your spinal cord, your nerves, brainstem, etc. It also is the functional unit of the brain and the nervous system. Therefore, when your brain sends electricity, or when your brain sends an electrical impulse, it's the neurons that are doing this function. The neurons are sending the nerve impulse or the electrical impulse. The spinal cord is sending the electrical impulse to muscles. So without neurons, you have no nervous tissue, and therefore your nervous tissue does not work. So the nerve impulses come from these neurons. Now, pretty much for the most part, neurons are very identical to all the other cells that we've talked about so far. If you go back to chapter 3 and you talk about all the organelles, the mitochondria and the Golgi apparatus and the ribosomes and the lysosomes and all those things, yeah, it's all pretty much the same. There really are two big differences with neurons compared to other cells. So as I mentioned, plasma membrane, the same, right? Every cell has a plasma membrane. Cytoplasm, yeah, we have a cytoplasm. You can see the nucleus over here in purple, right? Cytoskeleton, same thing. We have these proteins that make up the shape of the cell, and once again, shape determines function. Now, as I mentioned, there are two kind of weird different exceptions. The first exception is the cytoskeleton in a neuron is kind of specialized, and it makes these neurofibrils. Basically, the cytoskeleton extends off of the center portion of the cell body, and that's what we call neuron processes, or these spider leg looking things. Most of them are called dendrites, but this big, long, skinny one that I'm highlighting or circling, that's called the axon. And hopefully dendrites and axon and cell body, that should ring a bell, because we did talk about those in the tissues chapter. But those are what neurofibrils actually form. And they're just basically extensions or unique modifications of the cytoskeleton. The second thing that's different about these neurons compared to most cells in the body, or at least a good portion of cells, is the majority of them are amitotic, which means they don't go through mitosis very often. When you kill a brain cell, you kill that brain cell, it doesn't grow back. So that's why you want to protect your brain. That's why you have a skull, a real thick skull, bones that protect your brain. You also have a lot of connective tissues in there connecting it, and fluid connecting the brain. So a lot of layers of protective barriers to protect your brain from trauma. And that's why it's such a big deal when you get a concussion or when you whack your head really hard, because if you have nerve cell damage, that could lead to more serious things down the road if it continues to happen. Let's review the different components of the neuron, information that we've already talked about in the tissues chapter, but it serves as a good review. The cell body, generally the largest part of the neuron, I'm circling that in purple. That's just kind of this round centered area where we're going to have, as I mentioned, all the same organelles. We're going to have a nucleolus, we're going to have a nucleus, we're going to have Golgi apparatus, mitochondria, all these things. One difference is you'll see this thing called a Nissl body or Nissl bodies. And all that is the rough ER. It just stains a little differently in nervous tissue. So they have a special name for it. It's called Nissl bodies, but it's basically the rough ER of neurons. And everything else is pretty much the same. It's where we have these nerve processes, this extension of the cytoskeleton, is where it gets a little different. So we have two types. We have the dendrites, and the dendrites are by far a lot more numerous. There are all these extensions that I'm highlighting in red, sticking out of the cell body. And remember, the job of the dendrite is to bring information to the cell body. So in order of electrical flow, the dendrites are number one, and then they send the message to the cell body, which is number two in most cases. There are exceptions, but we're going to be talking about most common. And then we have the axon, and notice there's only one axon. We have multiple dendrites in most neurons, and only one axon. And the job of the axon is to carry the message away from the cell body to something else. And in the muscular chapter, or muscle physiology chapter, we talked about how the nerve carries it to a muscle. And then you can see in this picture where we have it talking to another nerve. Now the dendrites are a little faded, but I'll highlight them for you. So this is a nerve-to-nerve connection. And you can also see, if I clear some of this out up top, you can see that these dendrites, for this cell that I drew all over, is receiving messages from another axon. So it's just kind of a big game of dominoes, if you will. This neuron up here talks to this neuron in the middle of the page, and then the neuron in the middle of the page is going to talk to this neuron down at the bottom. And it's just kind of a game of Yeah, dominoes, or what's that game we played when we were kids? Telephone. You whisper a message, and the message has to go to the other side of the room. It's basically essentially what we're doing here. So when I was studying anatomy, and specifically neuroanatomy, as an undergrad and a graduate student, I never really understood the concept of how one nerve controls an entire muscle or even a muscle group. For example, in grad school, we actually worked on cadavers, human cadavers, and we would remove all the skin and all the fat and actually find the individual muscles. And what's really, really cool is along this process, you would find a lot of the big blood vessels. You'd find arteries and veins, and you'd grow an appreciation for how the circulatory system fits with other organ systems. But one thing that I always struggled with was how small the nerves were. The nerves were microscopic. Sometimes you couldn't even see them, or sometimes you accidentally sliced through them while you were dissecting, and you didn't even realize it. So as I continued to study and get a bigger picture of this, I finally learned how this works. And it was really late in my education, but I'm going to introduce it to you now. So one nerve, for example, if we take the femoral nerve in the femoral region, your upper leg region, your thigh region, one nerve is composed of hundreds and hundreds, possibly even thousands of these neurons. So that's one thing. And then another thing is these neurons can split multiple times. So in this picture, if you look at the picture on the right, I'm highlighting the axon in purple. That's the axon. Now in black, I'm going to show you a lot of times what happens with axons is we get extensions kind of at this 90-degree angle, and then they kind of do their thing throughout the axon. And what they call those extensions in black are axon collaterals. So basically the axon can split into 2, 3, 4 multiple types or multiple axons. So that's one way that we can cover surface area. You think of a really, really large muscle like the rectus femoris. How does a tiny microscopic nerve talk to the rectus femoris? Well, one, we have a lot of neurons and we have these axon collaterals. And then if you notice at the end of the axon collateral in the picture, you have other branches. So you have branches branching in the branches. So those branches are called telodendria. And those telodendria eventually turn into at the very, very end is what happens or what occurs as our synaptic knob. And that's what we talked about with the neuromuscular junction. If I draw my muscle cell right here, remember we had our muscle and then we had our big, what I call the axon terminal, same thing. Bad picture, but let's try that again. Remember, I kind of drew it like a triangle thing. And then we had the vesicles, right? And the vesicles had acetylcholine. That's what we're looking at here. We're looking at a very, very end part of a telodendria. And what that's called is the synaptic knob. And when some part of the synaptic knob is talking to something else, we call that a synapse. So that's what we're looking at here. With all the telodendria and all the axon collaterals, we can get a nerve having thousands and thousands of extensions all throughout the muscle. So when you go and straighten your leg or kick a soccer ball, it makes sense that that all happens very, very quickly and evenly because all of the muscle is innervated by that one nerve. Now, one more thing I want to point out on this picture is how these nerves are stimulated. You can see once again, we talked about this on the previous slide, but you have this synaptic knob or one of these telodendria talking to one of these dendrites. And if the message is strong enough, it's going to release acetylcholine. In most cases, it's going to release acetylcholine, which will start a new electrical impulse in this neuron. And then if it's strong enough, we'll send it down the axon and it will reach a telodendria. Here we go. Here's the synapse. So it'll reach this telodendria, which will talk to another dendrite. So on the next slide, we're going to confirm this order of how electricity passes through the neurons. So once again, the order is dendrites, cell body, axon, or DCA, I guess you can say. So in this picture, I can draw, you can kind of extrapolate. We have some cell body, we have a dendrite, few dendrites here, and then here's the axon that's connecting to the neuron in the middle of the page. So same thing, you have D, C, and A. And then down here you have the axon at the very bottom of the image. You have the axon talking to a few dendrites. And then we have a cell body, and that axon is going to go somewhere else. It can be another neuron, it can be a muscle, it can be a sweat gland. It doesn't matter. That's the order of electrical flow. Now that we understand the general anatomy of a neuron, let's talk about the different types of neurons throughout the body from a structural standpoint. So you can see in this picture we have three different shapes. They're all neurons, right? They all have cell bodies, they all have axon, dendrite looking things, but they are going to be a little different and mainly based on their location. So you have three types. You have unipolar neuron, you have a bipolar neuron, and then you have multipolar neurons. And what I want to talk about on this specific slide are the multipolar neurons. So what does all this polar stuff mean? It just means how many appendages or how many spider leg looking things are branching from the cell body. So with a multipolar neuron, what we're going to have is multiple branches. And that's what we've been typically talking about in the last few slides, where you have several multiple dendrites and typically only one axon. But even though we only have one axon, because we have many dendrites, it's a multiple number. And the reason why all of the pictures look like this with everything we've studied so far is because well over 99% of the neurons in your body are this type of neuron. It's a multipolar neuron. So you have 100 billion neurons in the nervous system, right? We saw that on the neurons slide. And what I didn't share with you is about 86% of that 100 billion, so 86 billion neurons are just found in your brain alone. And all of those neurons are what this shape, this multipolar neuron, a lot of dendrites, and typically only one axon. So that leaves only 14% for the rest of the body, and even that 14% is mainly multipolar. So on the next two slides, we'll talk about these other ones, but the unipolar and the bipolar have to share that 1%. So let's just say for easy purposes, the unipolar is going to be 0.5%, and then the bipolar will be the other 0.5%. So in terms of abundancy or how popular these neurons are in the body, multipolar by far the majority of your nervous system. So what a bipolar neuron is, looking at this picture on the right now, is we have a cell body and we just have bi meaning two, like a bicycle, right? So we have two branches. We have one dendrite and we have one axon. And the reason why this is so rare compared to other neurons in the rest of the body is these are really only found in some of your special senses. These are found in one part of your retina in your eye that helps you see. It's also found in a certain part in your inner ear, and then your olfactory pathway helping you smell, and that's it. It's really found in no other places in the body. So when you compare the eye, ear, and the smelling area to the whole rest of your head to toe body, it's a very small amount of neurons. And then last but not least, we have the unipolar neurons, and the unipolar neurons are also sometimes called pseudo-unipolar, because if you closely look at the structure, the anatomy of this, it looks very similar to a bipolar neuron, right? We have a dendrite type thing going one way, and we have an axon type thing going the other way. But the difference is what is connecting to the cell body. And if you notice, there's only one structure connecting to the cell body here. And then that one structure splits into one dendrite and one axon. So even though it looks bipolar, it is considered unipolar, or as some folks call it, pseudo-unipolar, because it appears unipolar, but not really, because we still have the same effect as a bipolar neuron. We have one that brings the information to the cell body, as an afferent message or as a more sensory message, and then we have one that sends it away from the cell body to the central nervous system. Now, where we find this is other sensory neurons, mainly going to be found in your skin. And yes, your skin is a very, very large organ, right? But compare it to all of the other neurons, the 86 billion in your brain, all the neurons in your motor nerves, your spinal cord, everything else, it's still a very, very small number. So typically these are going to be found in your skin, and this is what all of those Merkel cells and those Pacinian corpuscles and Meissner's corpuscles and free nerve endings that we talked about in the skin chapter, that's what these are. They get stimulated, they get the message, and they send it to the brain saying, hey, something's hot. Hey, something's touching me. And that's pretty much their only job. And then we can talk about neurons from a functional standpoint. So you're seeing kind of a theme here that we can separate the parts of the nervous system or even the neurons themselves from a structural, what they look like, or functional, what do they do. And believe it or not, the functional you actually already know. We've already discussed it. So if a neuron is bringing information into the central nervous system or into the spinal cord or eventually to the brain, is where most times it's going to go, it's referred to as an afferent neuron, right? Or an afferent message or an afferent nerve. So what we're doing here is something's touching your skin and we're sending it to the spinal cord. Now, sometimes the spinal cord doesn't really talk to the brain. It's just kind of what we call a reflex. But a lot of times, if something's hot or cold, it's talking to your brain. So the message goes to the spinal cord, and then the spinal cord will send it to the brain. And then the brain will send the message back down to the spinal cord, and then it will go out to the muscle to move your arm from the hot or cold environment. So if something's coming towards or sending a message towards the central nervous system, we call that an afferent nerve or neuron. If something is leaving the central nervous system, such as going to a muscle or going to an organ other than the brain, that's called an efferent. Remember we said A for arriving, A for afferent, and then E for efferent, E for exiting. So examples of those afferent sensory neurons, that's the really rare ones. That's the unipolar and the bipolar. So we talked about the skin, right? The ones in the skin are unipolar, and you can see it confirmed by this image right here. You have a cell body, and then it's that pseudo unipolar shape. So we have this kind of one extension, and then the extension splits into two. So one is coming from the skin, bringing information into the cell body, and then one is going away from the cell body, bringing it to the central nervous system, in this case the spinal cord. So that's where we saw that unipolar, that 0.5%. And guess what? That other 0.5% is also considered part of your afferent message, or your afferent neurons. So we talked about the inner ear. That's what helps you hear is these bipolar neurons get stimulated. They send a message eventually to your brain. Same thing in your retina. It's kind of a domino effect, but these bipolar neurons get stimulated, and they send a message to another type of neuron. That's going to eventually send the message to your brain to help you understand what you're seeing. So this is that 1%, these afferent neurons. It's the unipolar and bipolar combined. And then we have the efferent neurons. And the efferent neurons are going to be part of that 99%. They're going to be multipolar. And you can see in this picture that it verifies that. You have a cell body with a few, not a lot of dendrites, but a few. And then it has the axon that goes to the muscle. Now, from a functional standpoint, we have afferent, which is 1%. We have efferent, and we are missing one. What I have not told you yet is the connection points between the afferent and the efferent. So it's not as simple as this picture illustrates. Let me explain. What happens is if we were to draw a neuron in the bottom left corner, let's draw a pseudo-unipolar neuron, or a unipolar neuron in your skin. And what happens is it connects to a neuron in your spinal cord. And then let's draw the brain up here. This is how it really is organized. So we have a connection. The spinal cord sends that message up to the brain. And what I'm doing here is I'm drawing several neurons. And then the brain talks amongst itself, and the brain's going to say, hey, this part's doing this, and this part's doing this, and the brain has several different messages within itself talking to one another. And then eventually, once the brain figures out what we're doing, we're going to send the message back down to the spinal cord. And then the spinal cord is finally going to send its final message out to the muscle here. So what we have in this picture is a whole lot of neurons that I just drew you. We only have one afferent neuron, and that's this green one right here. And once again, the definition is something that's bringing information to the central nervous system. And our central nervous system structure is the spinal cord here, and eventually our brain. We only have one efferent neuron in this picture, and that's this last one, this purple one, that's leaving the spinal cord and actually going to the muscle. Okay, so then what is everything else? I drew like 14 red neurons in the middle between the spinal cord and the brain. What these are called interneurons, and this is the majority of that 99% of your body. So yes, these guys in purple talking to the muscles, they are multipolar and they are common, but you only have 500-600 muscles in your body, right? Your brain has billions and billions of neurons, and most of them are multipolar from an anatomical or a structural standpoint. From a functional standpoint, they connect to each other. They're constantly talking with one another, and what we call these are interneurons. We'll cover this more on the next slide. So here is an interneuron. So we have three types of neurons from a functional standpoint, right? We have the afferent neurons, which are very rare. They're the sensory. We have the efferent neurons, which basically just go and talk to the muscle or a few organs, also very rare. And then by far, here's the 99% from a functional standpoint. And they're called interneurons. They're the connectors. And it makes sense that if 99% of neurons are interneurons from a functional standpoint, they also belong to the 99% from a structural standpoint. So they're going to be multipolar in shape. Now, once again, what they're doing is they're connecting the afferent to the efferent. But it's not just one. In this picture, there's only one. If you look at the picture of the spinal cord, here in green is our unipolar neuron, and then it connects to a neuron. And then in purple is our efferent neuron going out to the muscle. And then connecting these two is the interneuron. But as I just showed you in the last picture, sometimes we're not dealing with a reflex. Sometimes we actually have to think and process what's going on. And in that situation, it has to go up to the brain, and then the brain has to send it back down, which would encompass a lot of neurons for that process to happen. So that is the majority of your neurons. It's figuring out, calculating what's happening in your body, right? So if we were to think about it from a functional standpoint, if I ask you what's 4 plus 4, hopefully you real quick you just say 8, right? But let's think about it from a functional neuron standpoint. You heard me ask what is 4 plus 4, and your afferent neurons in your ears turned that message into your brain and said, hey, you're being asked what 4 plus 4 is. And within milliseconds, your neurons are zip-zapping back and forth hundreds of miles per hour, calculating what 4 plus 4 is. Or if you don't have to calculate it, it's going directly into your memory bank in your brain and saying, hey, what's 4 plus 4 again? And then once it recalls that information, then it sends an efferent message to the muscles in your tongue and in your mouth for you to say 8, right? So there's a lot of processes happening. You heard what is 4 plus 4, you said 8, but in the middle of that, there was a lot of different things going on in your brain. And if you're multitasking, maybe you're playing on your phone while listening to this video, and when I said 4 plus 4, or you're writing notes, or you have the TV on, and your brain is processing multiple things all at once. And that's the interneurons that help you do that. So when people say you only use 10% of your brain, they are absolutely dead wrong. Your brain is using the majority of your neurons to do your multitasking, your everyday activities. It's just you're only probably using 10% of your brain consciously, focusing on one thing at a time. And it's because of your inner neurons that allow you to use more than 10% of your brain. Okay, so for now, let's stop our discussion on neurons. We'll come back to them when we get to the physiology part. Here, let's start talking about what protects the neurons, what keeps the neurons healthy. Because remember that the neurons are amitotic, which means once they die, they pretty much die. Now, there are some neurons in the PNS that can regenerate if damaged, but in the CNS, it's extremely dangerous to have dead neurons. Those aren't really going to grow back in most cases. So the cells that protect our neurons, that make sure that they're well supported, are called supporting cells. They're also known, they have many names, they're known as glial cells. They're also just known as glia. And on the next slide, you'll also see them known as neuroglia or neuroglial cells. So many different names, usually has the word glia somewhere in there. And we're going to have all sorts of different glial cells. We're going to have 5 that we're going to talk about, and I'm going to show you a chart with 6 actually. But you can see in this picture, it is a train wreck. It looks nuts, right? You have your neurons that you can clearly see. I'll highlight that in red. Here's your cell body, and then you can see your axons wrapping around. Here's another neuron over here. But they are all intertwined and jumbled up and connected with a whole bunch of other cells. Now, if you remember back to our histology lecture and lab where you looked at the pictures of nervous tissue, it kind of looks like this. It has 2 or 3 alien-looking spider leg cells, and then the rest of them is just crazy dots everywhere. And those crazy dots are these cells, the glial cells. And also notice the ratio. I said we had about 100 billion neurons in our body. We have 9 times that, almost 1 trillion glial cells. So 9 times the amount of supporting cells, once again, because the neurons are amitotic. So if these things die, we're in trouble. So we need to keep them alive, protected, supported in any way we can. And what accomplishes that are these cells, the glial cells. So there are a total of 5 that we're going to cover, as I mentioned, and we're going to go through them each on their own individual slide. So here are the 5 that we're going to talk about. In no particular order, we have the astrocytes, the microglia, the ependymal cells, the oligodendrocytes, and the Schwann cells. So before we dive into the details, I want to point out that the first 4 only are found in the CNS. They're only found in the brain, brainstem, and spinal cord. And that's it. The oddball one, the one that's remaining, the Schwann cells, these are only found in what's left, the PNS. Exactly. So most of these cells are only going to be found in your brain, brainstem, spinal cord, and then Schwann cells everywhere else. So let's talk about the astrocytes first. The astrocytes, really, really cool. It's hard for me to pick a favorite supporting cell, but if I had to, it would probably be these guys. They're really, really neat. So the astrocytes are, once again, they're one of those 4 that are only found in the CNS, and they get their name, astro meaning star, heavenly body looking thing, and they look like a star-like cell. And what they look like in this picture are the green guys. So you got this kind of circular cell body, and then they have these appendage dendrite looking things all over the place, and it makes them look kind of like a sun or a star. So that's where they get the name. So you can see there are 2 astrocytes in this picture. One at the bottom too. Out of those 900 billion glial cells, these are the most numerous. There are more astrocytes than any other type of supporting cell. They're also the largest. And their job is to connect the neuron to the bloodstream. Very, very important job. Bloodstream, big key theme here. So, once again, we need to make sure that these neurons are happy, healthy, working efficiently. So what we need to do is we need to get them a lot of oxygen and nutrients all the time. Well, where are we going to get those oxygen and nutrients from? You guessed it, the bloodstream. If you take a look at this picture, this little red tube right here, it's got some red blood cells floating in it. What these red tubes are called are, well, they're blood vessels, but in this case, they're really, really, really small blood vessels. And the smallest blood vessels in the human body are called capillaries. And it's basically between an artery and a vein. You can see another one here. So what the astrocytes do is literally they connect the bloodstream to the neuron. You can see the neuron. I just circled it in green. Let me circle it in black so you can see it a little better. These are the neurons. And notice they are connected by an astrocyte, which is connecting it to the bloodstream. Astrocyte in green, connecting it to the bloodstream. So what this does is it forms something called the blood-brain barrier. Remember, these are only found in the CNS. So the fact that it's found in the brain should make sense. What the blood-brain barrier is basically a very, very good filtration unit or a very, very good security network in the brain. So a lot of these capillaries throughout the body, think about the capillaries in your fingers or in your face, or in your legs. If you cut yourself shaving, usually, even if it's just a micro cut, you bleed for a little bit, right? And it kind of just oozes out and it just continues to bleed sometimes. That's because the capillaries near our skin are very, very leaky. And that's normal. That's how they're designed. They're designed to get a lot of oxygen and nutrients in and get kind of all the junk out. However, in the brain, we have to be really, really careful of that, right? Skin, not a big deal. We're constantly killing skin cells, making new skin cells. Who cares? In the brain, we really, really need to be careful of what's leaking out of our bloodstream and what's actually getting inside of our brain or nervous tissue. So if we have a toxin, if we have some type of aluminum or mercury or some kind of heavy metal, arsenic or copper or something that's floating around in our bloodstream, usually it will affect other organs before affecting our brain, because in healthy individuals we have this lockdown system of the blood-brain barrier where these capillaries don't really leak that well. They're kind of closed shut for the most part, and the only thing that is really able to access them and to get stuff out of them are the astrocytes, for the most part. So here's what the astrocytes do as they form this blood-brain barrier to make sure bad stuff doesn't get into our brain, but it also gets the good stuff into our brain. So it's going to bring oxygen, and I'll just put NUT period, nutrients, in, and it's going to get the waste products that our neurons make. All cells make some type of waste product, right? The waste products have to get out of the neuron. Where's a really, really good place to send waste products? Yeah, bloodstream, right? Because it's going to go to your liver, your kidneys, your lungs to eventually be processed or filtered or killed or get rid of. So exactly, the job of the astrocytes is to get oxygen and nutrients into the blood-brain barrier, into the neuron specifically, through the blood-brain barrier, and to get the waste products out so stuff doesn't accumulate in the brain. Very, very important. Our next type of glial or supporting cell is called the microglia. And before I start talking about the microglia, you can see this is a cool photo, cool illustration table showing you the different types of cells. We just covered the astrocytes in the bottom left corner right here. And you take a look at the microglia, they kind of look the same, right? They're just kind of this star-looking shaped cell. And we're going to talk about all of these except the satellite cells. But take a look at the chart and make sure that it's accurate. You have these four in the CNS, and then the only one that's found in the PNS that we're going to talk about is the Schwann cells, or are the Schwann cells, so that is accurate. So let's talk about the microglia. The microglia are small, that's where they get the name micro from, right? And once again, they're only found in the CNS. They're not found in the PNS. And normally they're stationary in brain tissue, which means they actually come from your bone marrow. They actually come as a white blood cell type deal, like a macrophage. And then what their job is to migrate to the brain. And then once the brain is happy with where they're at, once they find their station, they kind of anchor down and stay there forever until they die. So they're really just hanging out there, and as I mentioned, they're macrophages. So just in case something gets past the astrocyte and starts floating around in brain tissue, guess what? We have a second supply of defense. We have a backup supply that are ready to phagocytize or basically engulf, endocytose, bacteria, debris, or viruses or bad things in our brain. So if the astrocytes fail, we do have a backup. But if the microglia fail, then we're kind of in trouble. We need to make sure that these supporting cells are working properly. The next type of cell are called the ependymal cells, and the ependymal cells are really, really cool, really unique, because they have cilia. Now, when we talked about cilia in chapter 4 in the tissues chapter, we said it was kind of rare. Usually it's found in that pseudostratified ciliated columnar upper respiratory area, right? Back of the throat, kind of nose, nasal cavity area. But we also do have cilia lining our brain and lining our spinal cord area. So these cells are epithelial cells, or they're modified epithelial cells, and this is what they look like right here. They line a membrane. That's why they're called epithelial or modified epithelial cells. And what they do is, what do cilia do? Cilia filters things, right? Think about your nose hairs. That was kind of the analogy we made. Your nose hairs catch boogers, catch pollutants, right? Make sure that stuff's not going down into our lungs, which will give us pneumonia or some type of lung issue. And these cilia are doing the same thing, except they're filtering a fluid. And that filtering, or that fluid, excuse me, is called cerebrospinal fluid, or what some of you may know as CSF. Now we'll talk more about CSF and where it's made and the direction and the circulation, how it flows and everything next chapter. But for right now, just take my word for it. CSF is a very important fluid inside the brain and outside the brain. And it just basically supports it by giving it a lot of nutrients, and it also protects it from the inside out as well, because it's kind of this fluid barrier. It also is the same exact thing in the spinal cord. We have this fluid inside the spinal cord. We also have fluid outside the spinal cord. It's known as cerebrospinal fluid. So you make the cerebrospinal fluid 3 times a day. You make a whole brand new batch. So we got to keep this stuff moving. We got to keep it circulating through our nervous system. And in order to do that, we have to move this stuff, because gravity's not always going to be able to help us, right? If I'm lying down taking a nap for 8 hours or sleeping for 8 hours, gravity's not going to be able to move this fluid. So we need these little guys, cilia, to actually beat back and forth and move fluid in the proper direction. And that's the job of the ependymal cells. It's because we don't want that fluid getting stale, because it does have a job. It does have very important ions, nutrients in there. So we need to make sure it's circulating all throughout our brain. Basically giving our brain a big nutrient bath to make sure it has everything it needs. Okay, and then our final cell for the CNS is an oligodendrocyte. So remember, astrocytes, microglia, ependymal, and these cells are only found in the CNS. Only found in the brain, brainstem, spinal cord. So oligo refers to few, and what we're looking at in this picture is several neurons. We're looking at 3 axons, or at least 3 axon processes. So the stuff in yellow, that's an axon. So here's 1, 2, and then 3. And the oligodendrocyte is this blue-looking structure that's covering the axon, or insulating the axon. And what an oligodendrocyte does is it makes this covering, or this wrapping, and this wrapping is called a myelin sheath. So a sheath is basically like a casing or a covering. Like if you have like a holster, like for like a knife or a gun or like a sword, if you remove the sword from its sheath, you're removing the sword from its casing, is what a sheath is. So real life example for you. What this is, what myelin is insulation. So in my car, as I drive, I have a cell phone charger, right? And the cell phone charger, I mean, it's old. It's probably like 10 years old, but it still works. But the problem why it doesn't work really well is because it's really frayed. You can see the insulation, the white coating around the wire is all torn and just broken apart and chewed up, and it's just all messed up because it's been in my car for so many years, and it's filmed between the seats, and it's just been used. So you can actually see the metal wire inside of the charger, which is probably not that safe, right? And we know that if the insulation to that wire is destroyed, that electrical current may not work as well. And I know this for a fact because when I charge my phone sometimes, it doesn't always charge. It doesn't work. Or if it does, I go on a drive for 20 minutes and it went up 2%. So anyway, at the end of the day, I need to get a new phone charger. But I say all this to kind of hone in on this idea that the oligodendrocytes are basically the insulation around a phone charger. And the more insulation you have, the better the charger is, the better the protected, or the more protected that electrical signal is. So hit the pause button on the video real quick and just Google like huge mainframe cables, like Google headquarter cloud mainframe, or like mainframe database, or just big computer term where you have big cables, and you'll see how fat these wires are. These big machines and these wires that connect these supercomputers together to run entire businesses and corporations, they are gigantic. They are really thick wires. So that's going to be really, really important as we talk about the speed of a message. One of the criteria to make a message faster is make the tube faster so the electricity isn't stopped or resisted upon. And the other one is the more insulation you have, the more protected it is, and therefore the faster that message goes. So, with all that said, what is a good insulator in the human body? What do we have insulating some of our organs, some of our tissues that keep us warm, maybe give us a little bit of energy? And your answer, hopefully, is adipose tissue, or for short, I'll just say fat. And that's exactly right. That's what this stuff is. What is myelin? Myelin's essentially fat. And what we're doing is we're wrapping the neurons in fat. So they are, one, they're protected, and two, it makes the electrical current go faster. So there's going to be some neurons, as I mentioned, that send messages 200 to 300 miles per hour in your body. And then there's going to be some neurons that only have like 2 or 3 miles per hour in your body. So the ones that go 200 to 300 miles per hour are the ones that are going to have a lot of fat around them. The more fat, usually the faster that message goes. So that's what the oligodendrocyte does. It actually wraps the neurons in fat to make the message go faster and protect the neuron. Once again, though, these are only found in the CNS. We're going to move on to the next slide and talk about the Schwann cells. The Schwann cells are almost identical to these oligodendrocytes. The only difference, or one of the only differences, is where it's located. So as I mentioned, the Schwann cells, pretty much the same thing here. It forms the myelin sheath in the PNS. That's the difference. It's not in the brainstem spinal cord. It's everywhere else. It's in your eyes, it's in your mouth, it's in your arms, your fingers, your toes, your knee. It's everywhere except your brainstem, and spinal cord. So you can see in this picture, not the greatest picture, but it does have a lot of cool labels for you that I encourage you to check out. But in yellow, you have your cell body and your dendrites right here, and then this whole big picture kind of coming at us in the picture, that's the axon. And this blue stuff wrapping around the axon is the myelin sheath. That's the fat. And the Schwann cells are the ones making this fat. And notice that there's many layers of it. You can see 1, 2, 3, 4, maybe 5, multiple layers of this fat. And as I mentioned, the more fat that surrounds this tissue, the more protected it is, and the faster the message goes. So by now we can appreciate that if we have a whole bunch of myelin in the central nervous system, that myelin is made from the oligodendrocytes. If we have a whole bunch of myelin in the peripheral nervous system, that myelin is coming from the Schwann cells. Now, there are cases where we don't have myelin surrounding an axon. Or if we do, it's not a lot. The axons are kind of embedded in the Schwann cell, and it's basically just one kind of cover around it. It's really pretty much exposed or naked. Now, there is a reason for this. So let me once again show you the differences between the speeds of some of these neurons in our body. Some neurons in your body run 300 miles per hour, as I've mentioned a few times now. Some neurons in the body run very, very, very slow. Anywhere from 1 to 2 to 3, maybe to 5 miles per hour. Now, what would be the purpose of having a very slow neuron? Let's think about it. The 300 miles per hour exist in your brain. They also exist with all those interneurons and all those motor neurons, those efferent neurons going out to your musculoskeletal system. So when I move my fingers or when I'm moving my mouth, those messages are going from my brain to those areas 2 to 300 miles per hour. It's really, really fast. And this is why it's physically impossible to tickle yourself. Think about it. I'm an extremely ticklish person. If you touch my toes or my sides or my armpit, I'm going to freak out. But I can't do it to myself. I can't tickle my own armpit or tickle my own toes. And the reason why is, yes, even if I try to surprise myself, the messages are going to my hands 300 miles per hour. But guess what? The messages to your brain is also going 300 miles per hour, so you're expecting it. You know exactly where you're going to try and touch and tickle yourself. Versus if somebody else is trying to tickle you, even if your brain is expecting it, your brain can't pinpoint the exact specific location on your skin, nor the pressure or the sensation of touch on your skin. And therefore, somebody else can tickle you. So our computer, our brain, all those interneurons work extremely fast. So what would be the benefit of having a naked neuron? A 1 to 5 mile per hour neuron? Well, a great example of this is found in your guts and your stomach and in your intestines, right? We don't want the messages from our brain to our intestines going 300 miles per hour, because every time you ate a piece of food, what would happen? If your brain was sending these messages 300 miles per hour, it would say, digest, digest, go, 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 go. And as soon as you ate something and it hit your stomach, you'd be running to the toilet, right? And you would never absorb any nutrients. It would just go straight through you, literally. In the case of the digestive system, we want to slow things down. We want to take our time, because the longer that stuff sits in your digestive system, the longer we have to absorb the nutrients, all those good carbs and fats and proteins. So we want to have it slow, and that's why those neurons are what we call exposed or naked. Now, if you have a bunch of neurons that are naked or exposed or don't have any myelin around them, they're called gray matter. Now, we're going to talk a lot about gray matter in the brain and spinal cord next chapter, but for right now, all that means is the non-myelinated axons, or really anything else that's not myelinated, such as your cell bodies and your dendrites. They don't have myelin. So they also have this gray looking appearance. So in areas of the brain, if we have a lot of cell bodies clumped together, it's going to appear gray, and we're going to call that gray matter. But in areas of the brain where we have a lot of axons that are filled with those oligodendrocytes or protected by those oligodendrocytes, they are going to be known as white matter. Now remember, back to your histology chapter. Remember that myelin is adipose tissue. What does adipose tissue look like under the microscope? It just looks like fat, right? It's just a bunch of circles. It's just a bunch of big white pouches of fat, of lipids. And that's what we're talking about here. So if you look at myelin, it's just all of that whitey tissue, or whitish tissue. So therefore, in areas of the brain or the spinal cord or even in our nerves, if we have a lot of myelin in the area, it's going to appear a whitish color. So we're going to have white matter of the brain and gray matter of the brain. We're going to have white matter in the spinal cord, gray matter in the spinal cord. And what differentiates those two colors is how much fat we have, or if we don't have any at all. So we'll talk more about that next chapter. On this next section, we're going to be talking about the action potential. We're going to be actually learning a little bit of neurophysiology, and we're going to be doing a tiny little bit of math, throwing some numbers and how this all works. All right, on the next few slides, we're going to talk about the electrophysiology of neurons. And believe it or not, we've actually already talked a little bit about this in Chapter 3 with the cell. We talked about the membrane potential, the inside of the phospholipid bilayer being a little more negative compared to the outside. We talked about the sodium-potassium pump and how that regulates it. We talked about how we want a negative voltage inside of neurons and muscles to make sure that they're not spasming and freaking out all the time. So a lot of this will be review, but we are going to get a little bit more specific on the next few slides. Let's review what a membrane potential is. So recall back in your Chapter 2 and Chapter 3, your chemistry and cell, that when salts basically are separated or dissociated in water, they carry a charge to them. And we learned this, and we talked about this in the muscle chapter too, right? We always drew sodium as Na+, right? We drew chlorine as Cl-. We drew calcium as Ca++. And then we also drew potassium from time to time with a K+. So what these are called, when these atoms have these charges to them, they're called ions. Okay, and it can be a positively charged ion or a negatively charged ion. Now, inside of pretty much all of your cells, but really we're going to be honing in on nerve cells, and we already talked about muscle cells in the previous module, is there's a difference between the inside and the outside environment in the cells or of the cells. So if we take a typical cell, and this can be anything, this can be a neuron or a muscle or whatever. Inside of the cell, we have a boatload of potassium. Now, we do have a little bit of sodium inside the cell as well. I'm going to draw sodium a lot smaller than that big K, that big potassium. And we have all sorts of other things. We know we have calcium. We know we have phosphate groups. We have big proteins that are negative. And we have all sorts of different ions. But mainly, the main ion found inside of pretty much the majority of your cells in the body is potassium. Now, on the outside of the cells, floating around in that extracellular, that interstitial fluid, it's kind of flipped. It's opposite. We have a lot of sodium on the outside and not that much potassium. So I'll draw a big Na and then a little k. So just with sodium and potassium alone, we have a concentration gradient, right? We have an imbalance. Now, what I want to point out to you, though, is we have a lot of these negative proteins, a lot more inside the cell than outside, but also the main thing is we have a lot of sodium outside the cell. And what that does is that creates a more positive environment outside the cell compared to the inside of the cell. Now, it's not exactly saying that the cell is negative, it's just negative compared to its outside environment, right? Say we're playing a basketball game and you are beating me, let's say the score is 50-30, right? I don't have negative points. I have 30 points against you, but I'm down 20 points. You are beating me by 20, so you can say that I'm minus 20 because I'm losing by 20 points. It's that same kind of principle. So what we have here is a difference, and that difference is regulated by that sodium-potassium pump because we want this difference. We want it to be negative inside the cell because that means the cell is at rest. Once again, you don't want your cells, your neurons, to be at what we would call equilibrium or homeostasis. Because if everything is even or zero, that means the nerve is firing. And if every single nerve in your body and every single muscle in your body is firing at the exact same time, you're not going to be able to do anything. You're going to be having seizure-like activities, and you're going to just be constantly spasming and twitching all over the body. So we have to rest. And what keeps us at rest is this difference that there's more positive ions outside the cell than inside. If you recall back to last module when we talked about the muscle chapter, what causes the muscle action potential? Yes, it starts with the calcium and the acetylcholine in the neuron, but it's the sodium rushing into the muscle cell, rushing down those T-tubules, that is the actual action potential or the electrical impulse of the muscle cell. So the fact that we have more sodium on the outside compared to potassium on the inside, it's when the sodium rushes in is what's going to cause the neuron or the muscle to fire or to send electricity wherever it needs to go. So we call this difference a membrane potential, right? There's an electrical charge and it's different, and this is stored up energy. Those sodiums want to go into the cell. Those potassiums want to get out of the cell. And we have this concentration gradient. And what this is measured in the human body is millivolts. And usually we abbreviate this as a little m and a big V. And volts are just a unit of electricity, right? If you change a light bulb, I'm looking at it right now. I'm looking at a 9-volt, I think it's 9. Let's say it's a 9-volt battery in my kitchen area right now. That's a lot of volts. That's a lot of millivolts, right? That would be 9,000 millivolts. So since the cells are microscopic, the body's really, from a microscopic standpoint, the structures are really small. We call them millivolts, so 1,000th of a volt. And we're going to be doing some math, as I mentioned, with these millivolts in just a moment. So let's move on to the next slide, and I'll show you another illustration of what I'm talking about with this concentration gradient, or this difference between the inside of the cell and the outside of the cell. Okay, so let's begin by getting our bearings, our orientation right. What we're looking at here, kind of in the middle towards the bottom, is a phospholipid bilayer. Remember, we have 2 layers of these phospholipids, and the lipids are the tail-looking structures that I'm highlighting currently in black, and these are the fat. This is the hydrophobic component, right? The part that doesn't like water. And then the circles that are actually touching the water, that's the hydrophilic part of the plasma membrane. So we're looking at the outside part of a cell. In this case, we're looking at the outside of a neuron, part of, let's say, the axon specifically. Okay, so inside, or at the very, very bottom of the page, notice you'll see the cytosol. And recall, the cytosol is just the watery stuff inside the cytoplasm. So that's the inside of the cell. And then outside is the ECF, or the extracellular fluid, also known as interstitial fluid. Now notice, along the edges of the phospholipid bilayer, you have these charges. Right, towards the cytosol, you can see there's all these negatives, negative, 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 negative. And on the other side of the extracellular fluid, there's all these positives, positive, positive, positive. Now, once again, don't be misled that the entire cell is negative. It's not. It has a lot of positively charged potassium ions inside. It's just, going back to that basketball score, it's just we have a lot more positive ions on the outside compared to the inside. So let's just go back to that score just to make it easier. So the outside has 50 points at halftime, and then the inside has 30 points at halftime. Both can be positive. It's just what we're doing is relative to one another, we're comparing them. So the outside is more than the inside. So what we could do is, instead of saying 50 to 30, we could say that this is 0, or this is normal, and then this is down by 20, or something like that. So the inside of the cell, compared to the outside, has a negative voltage, outside has a positive voltage. And what we're looking at in this picture is we are actually measuring that difference, that concentration gradient, or that imbalance. And we're using a voltmeter. Once again, we're measuring things in volts, or in the case of the human body, millivolts. And this number, -70, is what we call our resting membrane potential, and you're going to see that on the next slide. This is what our neurons are at rest, when the neurons are not firing electrical messages, when we're not communicating with other muscles or neurons or sweat glands or whatever we're talking to. It's chilling at -70. Now, as soon as the brain or a different part of the brain wants to fire, wants to send an electrical message to your finger, what's going to happen is all of these positive ions, or at least a huge portion of them, are going to rush into the cell. And when that happens, this voltmeter is going to jump from -70 all the way to about +20, well, +30 to be exact, right here. So we're going to have a huge change in that voltage when those ions change their position. But for right now, what's keeping them at bay, at rest, mainly is our sodium-potassium pump that we're going to show you in just a moment. So this slide, we're talking about the resting membrane potential, the RMP, and remember our gold standard, our number that we're going to always use is -70 millivolts. And what that means is this is when the neuron is not doing anything. It's not sending electrical impulses anywhere. It is chilling. It is resting. And the only reason why this is occurring, or what this definition actually means, is it's just the outer surface, that extracellular fluid outside the cell, is a little bit more positive compared to the inside of the cell. And what this membrane, or what this, we're going to add a term real quick, this term polarized. What that means is basically there's an imbalance, there's a concentration gradient. And we're going to add a few prefixes to this term. We're going to say depolarized and repolarized in just a little bit as we change that concentration gradient with stuff moving in and out of the neuron. So for right now, polarization, it just means differences, okay, changes or an imbalance. Something's lopsided, something's not even, so it's polarized. If you take a look at the picture at the bottom, you can see what we have going on is a concentration gradient. We have 6 structures inside the cell. We have 1 on the outside of the cell. And we have a protein channel. Remember that a lot of things that transport in and out of the cell have to use protein channels to do that. Some can't just move right through the phospholipid bilayer. So you can see that in this specific case, it's called a voltage-gated channel. So it's excited by electricity. And you can see this kind of ball and chain mechanism blocking the ions from moving on the left side of the picture. And then if you look at the right side of the image, you can see the ball and chain structure is moved out of the way. So we can start having diffusion where this high concentration stuff is moving to the low concentration. And that's going to change that concentration gradient. So what's keeping our -70 at -70? What's keeping this RMP stable? Well, there's a lot of things, as you can see in your notes. First off, those large proteins that are negative, remember I drew those negative, that blue negative sign inside the cell. Those proteins are too big. They can't just leave the cell by themselves due to a concentration gradient. So they're stuck inside the cell, which makes the environment a little more negative, right? Those sodium channels are closed unless we activate them, unless we are trying to start an electrical impulse. So let me draw you that representation of the cell again. Once again, we do have sodium inside the cell, but just not a lot. I'll draw a little Na. But we do have a whole boatload of sodium outside the cell, and that's why I'm drawing the big Na on the outside. So a lot more sodium on the outside compared to the inside. This is a concentration gradient. That sodium on the outside wants to get inside the cell to balance things out. But as I just mentioned, they're closed. Those channels are closed. That can't happen. We also have the sodium-potassium pump working for us. Remember what the sodium-potassium pump is from chapter 3. What we're doing is we're kicking 3 sodiums out, but we're only bringing in 2 potassiums. So I'll put 2K right over here. So we're just further increasing this concentration gradient. We're making sodium more prevalent on the outside, and we're making potassium more dominant on the inside. But look at the net overall charge, what we're doing here. We're losing 3 positive ions, but we're only bringing in 2. So I gave you $3 to go buy, I don't know, a candy bar or something, right? I gave you a $3 loan expecting you to pay me back, and you only pay me back $2. Well, I lost $1, right? I gave you 3 left my wallet, but only 2 came back in my wallet. So at the end of the day, I lost $1. And that's the same thing here. We have a net -1 charge. So as stuff is moving back and forth in and out, we're keeping this thing negative based on all of these different factors. You can also see that potassium kind of has some flexibility of moving around that concentration gradient, but sodium generally can't. So what this does is it keeps that positive charge on the outside of the cell, and it keeps us stable at that -70 on the inside of that cell membrane. Here's a review of the sodium-potassium pump. Once again, as I just mentioned, we're kicking out 3 sodiums and only bringing in 2 potassiums. Remember, this is a form of active transport, so we are using ATP. This doesn't happen without ATP. And you can see that the orange little hexagon shapes are sodium. So with the help of ATP, down here, what we're doing is we're kicking out 3 of these sodiums, and then we bring in 2 of the yellow oval looking structures. We're bringing in 2 potassiums. And that changes the conformation or the shape of the protein, and then we just do that over and over as long as we have ATP. Okay, on this slide, we're going to just talk about an overview. The next several slides are going to talk about these 5 points in more detail. So generalizations about the nerve impulse. First off, what we're going to do in just a moment is we're going to change the terminology of nerve impulse. We're going to turn it into something called an action potential. I know we've been talking about nerve impulses for a while, but an action potential is a little bit more accurate, and I'll show it to you on the slides. So first off, a stimulus initiates the nerve impulse or the action potential. Something has to happen, right? Pretty much for the most part, at -70, we're at rest. Those proteins can't move out. The sodium-potassium pump is regulating things. Sodium can't rush in. So we're stuck at -70 unless something changes, unless something touches us, unless our brain wants to initiate a movement or a thinking process. So something has to happen in order to get this ball rolling. Just like, remember when we talked about with the muscle contraction, in order for the calcium to rush into the cell and the acetylcholine to be released in the muscle or in the neuron to the muscle, we have to have something start in the brain. It all starts in the central nervous system. Number 2, the nerve impulse or the action potential is electrochemical. Remember, between the neuron and the muscle is a space. We have that synaptic cleft, and that acetylcholine gets released. The acetylcholine is a chemical. It's what we call the neurotransmitter. So it's not just this big electrical circuit all throughout your body. There are places in the body, such as your heart and such as parts of your brain, where it is electro, or electro, where it's just this continuous wiring network. But for the most part, the majority of your body is electrochemical. There's an electrical message that ends up in resulting in a release of a chemical, and then that chemical starts a new electrical message. And then that ends in a chemical release, which starts a new electrical message, and it just goes electrochemical. We'll talk about that as well. Number 3, the electrical part of the action potential flows in a wave-like fashion along the plasma membrane. It kind of, basically, if we're running from Florida to New York, you can't, or flying from Florida to New York, you can't just get there. You have to go from Florida to Georgia to South Carolina, North Carolina, Virginia, and work all your way up. So yes, you have to start at A until you get to point B. Number 4, the nerve impulse speed varies due to how big the axon actually is, how thick the tube is, and how much myelin it has. We talked about that. The more myelin, the faster and more protected it's going to be. And then last but not least, number 5, the action potential or nerve impulse does not lose strength from start to finish. We're going to have something called the all or none principle that we're going to introduce in just a moment. But there's also sodium channels all throughout the axon that make sure that we don't run out of this positive electricity flowing through the inside of the cell. So now that I gave you kind of an overview of these, let's dive into the details of all 5 of these. So we'll start with number 1 and just work our way through. Number 1, a stimulus initiates a nerve impulse, and you can see here in the notes that action potential is introduced. This is the same thing as a nerve impulse. Action potential is just a little bit more accurate, speaking in electrophysiological terms. So something has to happen, something has to excite your body in order for this process to be rolling. Think about the unipolar or afferent neurons in your skin. Right now, unless you can really psych yourself out, if you just look at the back of your hand, the back of your hand is not telling your brain anything. There's no information about the back of your hand in communication with your brain. But if you start scratching the back of your hand, those unipolar neurons get stimulated, and they send the message to your spinal cord, then up to your brain saying, hey, you're scratching your skin. Right? The stimulus is you scratching. Something is touching your skin, which is exciting those neurons. So something has to get the ball rolling. Now, once the ball gets rolling, take a look at what happens. Sodium channels open. Now remember, inside of our cell, that big concentration gradient, if I draw it once again, we have a little bit of sodium inside the cell, but we have a boatload outside the cell. So we have an imbalance. If sodium channels are open, which way are they going to flow? Are sodium ions going to flow from outside to inside or in to out? And what you should be answering, hopefully, is the high concentration is always going to go to the low. So sodium is going to start rushing into this neuron, into the cell. And when that happens, you see what's going on in the chart. We're at -70, followed my red line, -70, and then as soon as those sodium channels are opened, we get this huge insurge, or this huge influx of sodium ions rushing into the cell, and we get this huge change in our millivolt measurement. So with that said, let's talk about this chart and what's going on here. We have a few different terms. Notice your x-axis is just time. This is actually time in milliseconds. It's very, very, very fast. Okay, it's not like this takes hours or something. It's thousandths of a second we're talking here. And then the y-axis, up and down, is just the millivolts. It's measuring how many millivolts. And you can see here's our resting. Here's our -70. I'll put RMP for resting membrane potential there. Okay. So, notice at -55, there's this dotted line running across the entire graph. What we call that dotted line is the threshold stimulus, and I'll do my best to spell this for you. It's just hard to do this with my mouse. Well, you know how to spell stimulus, but threshold. And then I'll just put stim. And the magic number, the universal number for our threshold stimulus is -55. What this means is if the stimulus is excited enough, or if it causes a big enough excitement, and it gets to that -55, we're going to open up the floodgates and we're going to have an all or nothing phenomenon. Which means once we hit this -55, we're going to shoot straight up to this tip top of the mountain. Right? And it's the same, kind of the same principle, the all or none principle of the muscle contraction. Remember, when a muscle fiber contracts, it contracts to its fullest ability, 100% pedal to the metal. Same thing here. If we hit that -55 voltage, it will go all the way up to the tip top. Now, if that doesn't happen, let's say we have a stimulus, let's say that there's a mosquito on the back of your hand, but it's really, really light and it's not stimulating your skin, those neurons in your skin, so your brain's not aware of it, and that's how the mosquito gets you, right? They stick their little thing in you and suck out your blood, and then you got this welt on your hand. It's because your brain didn't pick it up, because your hand neurons weren't stimulated. And they also, I've been told, they also have weird chemicals that deaden your skin. I don't know about that. Probably true. But anyway, let's talk about human physiology here. So if I have something, let's start here at -70, follow my red line. If I have something stimulate my neuron, and it goes up to -65, -60, but it never hits that -55, what's going to happen eventually is it's just going to come right back down to -70 and even out. Because remember, that sodium-potassium pump is pretty much working around the clock to maintain that resting state, that -70. So unless we excite it enough, nothing's going to happen. All right, are you with me so far? Hopefully yes. If not, I don't know your question. So rewind to the beginning of this slide, rewind about a good 2, 3, 4 minutes, and just re-listen to what we're talking about here. Play it slower. Okay. So now let's talk about the numbers. How many millivolts do I need in order to reach my threshold stimulus? So if I start at -70 and I have to climb to -55, there's a certain number that I need to get to. So if I climb 5 millivolts, I'm at -65. If I climb 10 millivolts, I'm at -60. So your answer is 15 millivolts. So in order for us to get from -70 to -55, or if you want, you can just get rid of the negatives. Just do 70 subtract 55. 70 minus 55 equals 15 millivolts. So if something is around you, if something is stimulating you, but it doesn't hit that 15 millivolts, it's not going to stimulate your brain. And the example I use once again, the mosquito or the ant bite, right? I got bit on my ankle a few weeks back, and I didn't realize it until the bite was already there. I didn't see anything bite me. And the reason why is because that 15 millivolts didn't happen. That ant is so light and I had other things going on. My brain wasn't paying attention to it. I didn't get that message that, hey, there's something crawling on you, flick it off. So once it bit me and it started itching, that's when I got that threshold stimulus and those pain receptors started becoming more activated in my brain. So 15 millivolts is our magic number. Once we hit that 15, we get something called the all or none phenomenon. And what that means is we're going to go from that -70 where we started, and we're going to go all the way up to +30. What we call this is depolarization, this upward swing of influx of sodium ions is what's causing this. When we're at -70 and we have a whole bunch of positive ions rush into the cell to the point where it's changing that resting potential, we call this depolarizing or depolarization. Okay, one more math equation for you here. If I start at -70 and we go all the way up to +30, from rest all the way to the tip top of the mountain, the peak, how many millivolts did we move? Or what's the total change of voltage? So from -70 to 0 is 70, and then add 30, what your answer should be is 100 millivolts. From the very bottom of this graph at -70 all the way to the tip top at +30, there's a 100 millivolt change happening. And this happens no matter what. Whether we are blinking our eyelids, something real easy that we take for granted, or we're lifting 500 pounds off of the floor. It doesn't matter. This is the exact same thing all the time. Now the difference is we're going to be sending a lot more of these -70 to +30 if we're lifting that heavy 500-pound piano. and we're going to be sending it to more muscle fibers as well, right? So we're going to have more activation or recruitment of muscles in order to do that. But the actual anatomy of the action potential doesn't change. It goes -70 to +30 no matter what we're doing. As long as we hit that threshold stimulus, it's going to happen. But everything that goes up must come down, right? Because we want to be able to do this multiple times. So as soon as we hit that +30, we start resetting. We start coming down. So what happens is we close those sodium channels, so no more sodium can come into the cell, and we actually open potassium channels. And what's going to happen when we open potassium channels? Remember our concentration gradient for potassium. If I draw a cell right here, we had a lot of potassium on the inside, not a lot on the outside. So big K, little k. Which way is potassium going to flow? It's going to leave the cell. And remember what kind of ion potassium is. It's a positively charged ion. So a little plus right there. So if we're having a lot of these pluses leave the cell, that inside is going to get negative again. So as we go through this huge negative, or this plunge toward negative, what we call that is repolation. We're repolarizing or resetting the cell back to normal. And then last but not least, you get this kind of extra dip, and that's called hyperpolarization. And that's just because the potassium ions, as they're leaving fast, and it actually goes below -70 to around -80 sometimes, even -90, and then it'll rebalance itself as we maintain that sodium-potassium pump and potassium ions start floating in, and it eventually just kind of balances out. But it just buys the neuron a little time to actually totally reset before we initiate another action potential at this -70, and then we can do it again. So make sure you review this slide, make sure you know the numbers, make sure you know the terms. Depolarization is when the neuron is getting more positive, going towards that +30. Repolarization is where we're resetting. We're going from +30, trying to get back down to that resting state. And then hyperpolarization is just kind of that in-between where we're going a little below -70. It's because these potassium ions are just leaving too fast because of that concentration gradient, and then we'll balance it out as soon as we kind of hover around that -70 area. Moving on to generalization number 2, the nerve impulse is electrochemical. This should be a review. We've already covered the electrochemical aspects of the neuromuscular junction. So picture, in the picture on the right, that this is the axon terminal, or the nerve, and then down here is the muscle, and everything's the exact same. The action potential has to travel down the neuron, right? And then what that's going to do is that's going to activate calcium channels to let acetylcholine eventually be exocytosed. Acetylcholine is going to bind with receptors on the membrane of the muscle to allow sodium down the T-tubules. It's the exact same thing here. The only difference in this picture is instead of it being a muscle, it's a neuron. But it's the exact same thing. If you notice at the very edge of the second neuron right here, those are receptors for the acetylcholine. You can see the acetylcholine, the little specks, the little dots inside the synaptic cleft. So everything's the exact same. Now, one thing I do want to point out is the terminology of a pre and a post-synaptic neuron. So remember, the synapse is just this connection or this communication. It's a neuron talking to a muscle or a neuron talking to another neuron. That's a synapse. So when we have this relationship, we can say that this neuron up here that's delivering the acetylcholine to the next neuron, this is called the pre-synaptic neuron. And therefore, the one receiving the acetylcholine and starting the new nerve impulse, that would be the post-synaptic neuron. And then eventually, this one would become a presynaptic neuron if it talks to another neuron. And pre-post, so on and so forth. It just makes a big domino chain. But everything else is the exact same. So if you don't know what I'm talking about, if you're not following along here, no worries. Just go back to the muscle physiology chapter, which is chapter 10, and spend some time on that and cover all of the nitty-gritty details of the neuromuscular junction. Number 3, nerve impulses or action potentials don't lose strength. And the reason why is because they're electrochemical. The neuron only, the message down the neuron only has to travel so far until the electrical message is stopped, and then the chemical takes over, the acetylcholine here in the synaptic cleft that I'm circling, which starts a new electrical current. So if it was strictly electrical, what would happen is the electric current would stop with these gaps, all these synaptic clefts. Basically, you're trying to charge your laptop or your cell phone, and you cut the wire 2 or 3 times. Well, no matter how many times you plug it in the wall, it's not going to work. The thing has to be connected. So, because this is an electrochemical type of connection, that chemical release, the acetylcholine, starting the new action potential down the postsynaptic neuron, is always generating that threshold stimulus, so it's always getting to where it needs to be, and it doesn't lose strength. We're going to talk about another feature of why the action potential doesn't lose strength in just a little bit on generalization number 5. But as you can read, if it was continuous, we would have a problem because of that synaptic cleft. It's not continuous. So we have to release a chemical to start the new electrical impulse, and then another chemical, and then another electrical impulse, and so forth. Number 4, the nerve impulse speed. So there are 2 main criteria that determine how fast this message goes. So remember, I gave you 2 drastic examples. I said that some neurons in our body go 300 miles per hour, right? I mentioned the tickling analogy, why you can't tickle yourself. And then I gave you the other extreme, the really, really slow neurons in your digestive organs. So the 2 main criteria is how big the tube is, the diameter of the axon, and what we've already talked about, the myelin. How much myelin do we have? So let's talk about number 1 real fast. I'm going to draw 2 tubes, and these tubes are axons, and they're going to carry electricity, because that's what they do, right? So we have tube number 1 in red, and then we'll have tube number 2, let's do green. There we go. Okay, so yeah, my lines aren't exactly straight, but you get the point of the exercise here. There are 2 tubes. The red one is a lot tinier. It's a lot smaller, right? And the green one is a lot bigger. Now picture electricity actually going through these tubes. When the electricity goes down tube number 1, or the red tube, it's going to meet resistance. It's going to bounce up and down and against these walls, which is going to slow down that message, right? If we're going down a lazy river and you keep bumping into the walls, you're not going to get to the end of the lazy river that fast. But if you look at tube number 2, the green one, if you have more space, you're just going to float right down the lazy river. You're not really going to be bumping into too many walls. You're just going to be doing your thing. And therefore, you're going to get to the final destination faster. And we're going to talk a lot about this principle with blood flow as well, with blood pressure and resistance next semester. So the bigger the tube, the less resistance, and therefore the faster the speed. And then, of course, how much myelin we have. And we'll talk more about this saltatory conduction on the next slide right here. Okay, and then this is our last generalization. Number 3 and number 5 got a little flip-flopped, mainly because they're the same thing. They kind of overlap each other. So if you go back to our main generalizations, number 3 says that the nerve impulse moves in a wave-like fashion. Well, yes, of course, it has to, right? If we start looking at this picture, if we start at the cell body area, which is up here, the very top right, the message has to move down the axon. So you can make the argument that's a wave-like fashion. But we also have gaps in the myelin in a lot of places in the nervous system, and that makes it kind of this jumping looking appearance I'm about to explain now. So with the myelination of axons, we have this phenomenon called saltatory conduction. And what saltare translates to in, I forgot, Latin or Spanish. I think Latin it means to leap, and then Spanish it means to jump, saltar, saltale, or something like that. But it means leaping or jumping in one or two multiple languages. Now, what is actually happening? What causes this jumping sensation? Well, let me draw you my own axon and I'll show you. So here's my cell body and dendrites. And then there's my long axon, and then covering the axon or protecting the axon, we have myelin. Okay, so these gaps that are found between the myelin are called the nodes of Ranvier. It's a French term, French word, so pronunciation's a little weird. Nodes of Ranvier. And basically what those are exposed areas of the neuron where there is no myelin, but there is something to help us make sure that we don't lose that signal strength. Because if you think about it, remember, the more myelin we have, the faster the message goes. So it would be kind of silly if we had this phenomenon where following at the bottom of the picture in red, the message is really, really slow and then fast through the fat and then slow and then fast and then slow and then fast and slow. And what would happen is we could maybe eventually it's so slow that we actually stop. But that's not the case. What's happening at every single node of Ranvier is there is a sodium influx. A lot of sodium ions are flooding the axon. So basically what we're getting is kind of like a bunch of gas stations. Say that we're going on a long road trip and the beginning of the road trip starts at the cell body and we're going all the way across the country to the end. And in order to do that, you have to stop, right? Or you have to at least have to slow down and get some gas. But at every gas station, you're charging your car with more gasoline, so you keep going and going and going. And that's essentially what we're doing here with the nodes of Ranvier, is we're creating new action potentials. So as the nerve impulse travels down the axon, we get this kind of wave-like fashion, but really we're generating kind of a new action potential at every single pit stop or gas stop at the nodes of Ranvier. So what they are basically are these glorified sodium channels. And at every single time, every single stop, we're going to flood the axon with more sodium so it keeps on moving faster and faster and faster until it gets to its final destination. And this is a slide just kind of giving you a different analogy. So I gave you the analogy with the gas station. Another one would be a staircase, right? If we're running up down the stairs or something like that. So I'll draw you a staircase here. And if you walk and touch every step, right, it would take you about 6 steps. 1, 2, 3, 4, 5. It would take you 6 steps to get to the top. But what if you were to run up the stairs and skip the steps, or skip a few steps? So if we were to run, we would hit 1, 2, and we would get upstairs in 3 steps. And that's essentially what saltatory conduction is doing. It's basically just skipping a step or making sure that the action potential doesn't die. So the reason why they call it that, to give you a bigger picture of that axon that I drew, here's my cell body dendrites axon, and then here's the myelin covering it. Essentially what we're doing is through the myelin, the signal is very, very fast. Remember, all that insulation makes the signal very, very quick, and it slows down maybe just a little bit, but all we're doing is just supercharging it with more sodium ions. So we're going to get into skipping mechanism over the myelin sheath cells. Now, it's not really skipping. It just has this kind of skipping appearance or the skipping idea because of the myelin. But in reality, the electrical signal is just going through the tube. And at every single pit stop, we're just filling it up with more sodium ions. And remember, all we're talking about when I say filling up with sodium ions or an influx of sodium ions, all we're doing is just depolarizing that part of the axon. So if we're at negative 70 millivolts, and we're flooding that part of the axon with a bunch of positively charged sodium ions, what's going to happen to our flat line? It's going to start depolarizing. It's going to go up. Exactly. That action, and then repolarization, hyperpolarization, and go back down to normal. So that's what we're doing there. So yes, it may slow down. The more myelin you have, the faster it's going to go. But in cases where we don't have myelin all throughout the axon, where there's these little gaps and the gaps are once again called nodes of Ranvier, we get this skipping mechanism, saltatory conduction, where we're generating kind of multiple action potentials all throughout the axon, so that signal never loses strength. It's going to get to its final destination no matter what. Okay, just a little bit more information on the action potential. A lot of this is review. Once again, the action potential is just another way of saying nerve impulse or electrical signal in the nerve. And we have to have a strong enough stimulus in order for this to occur. And remember, that strong enough stimulus was called that threshold stimulus. And that magic number for the threshold stimulus was negative 55 millivolts. So we needed to go from negative 70 to negative 55 in order to accomplish this. And then as soon as we hit that, the floodgates open. What happens is sodium ions open. So sodium channels open, and when sodium channels open, a lot of sodium floods into the cell, and that's what's causing this huge spike, this huge increase of what we call depolarization. Now on the next slide, we're going to cover repolarization, and we're going to talk about what happens with that process of the action potential. So with repolarization, what's happening is we are closing the sodium channels. So at this point right here, when we get to the top of the mountain, sodium channels close, so no more sodium can enter the cell, and potassium channels open. Now let's think about that real quick. Once again, the main ions found inside of your cells. Potassium is the predominant ion, or positively charged ion in the cell. Very little potassium on the outside. So when we open potassium channels, which way are they going to flow? Well, they're going to flow from high concentration to low concentration, so they're going to flow out. And what's going to happen to our millivoltage if positive ions start leaving the cell? Well, it's going to start going down. It's going to repolarize. So two things are causing the repolarization. Mainly, the sodium channels are closing, so no more sodium can get in, and the potassium channels open, so potassium can leave. So we get this double whammy, which brings us closer to that 0 and then that negative 70 pretty quickly. Now we're also doing the sodium-potassium pump, once again trying to get us back to negative, right? We're kicking out 3 positively charged ions while only bringing in 2, so that's going to help with this repolarization process as well. And as you can see with this last bullet point, the repolarization occurs along sections of the neuron just behind the action potential. So just like the action potential is going through those nodes of Ranvier in a wave-like fashion, picture going to a football or soccer game and you do the wave, right? As the electrical message goes in this wave-like fashion, so does repolarization. So if you're doing the wave and you just stood up and did your wave thing, then you sit down, right? And the person next to you stands up and then they sit down, and it's the same phenomenon, the same principle. And then our last topic before summarizing all this up is the refractory period. And the refractory period is basically this time frame where you cannot create another action potential, or if you can, you have to go a little above and beyond what's normal. So let me explain. As you look at the picture and you see our typical action potential, What happens is you go up to positive 30 with our depolarization, and you have to reset. When you come back down, you have to go all the way down to negative 70. You can't go halfway and then create another action potential, or go even further down and then go higher than positive 30. It doesn't work like that. Once again, it's an all or nothing thing. So it's going to have to go all the way down to negative 70. And if we hit that threshold stimulus, it goes back to that positive 30. So the refractory period is, can we re-stimulate this thing so we can make multiple action potentials? So for example, if we are to go positive 30 and then all the way down, as soon as it resets, can I make another one? And then as soon as it resets, can I make another one? And can I make another one? Can they be back to back to back to back to back? Yes. It just has to have a strong enough stimulus. As long as we're hitting that threshold, this can happen. Now, the time frame that it can't happen is something called the absolute refractory period. And as you can see in the definition here, another action potential will not begin, no matter what you do. And the time frame when this absolute refractory period occurs is during the depolarization state. So when those sodium channels are open, no matter what we do, even if our brain is saying keep going, keep going, keep going, no matter what stimulus we do, when we hit that negative 55 and the sodium is rushing in, that's it. You can't create another one. There's nothing else you can do. You have to wait until those sodium channels close and we start resetting before we can even think about making another action potential. So it's kind of on lockdown. Nothing's going to work. The time frame where you can generate another action potential as soon as you come to rest is called the relative refractory period. And as you can read in the definition, another action potential can be initiated if a stronger stimulus is present. And the relative refractory period happens during this repolarization phase. So this is when the sodium channels are now closed and the potassium channels are open. And what this is saying, or what this is trying to show you, is if we want to make another action potential immediately following this one, we can, but we have to stimulate the neuron stronger than before. So remember, how many millivolts do we need to go from rest to our threshold stimulus? So in the bottom left corner, if you look at the numbers, negative 70 to negative 55 is what I'm asking. Your answer should be 15 millivolts. That's all you need for at rest. But if we want to stimulate another action potential, as soon as we come down the rest, do this and make another one and do this. What we have to do is we have to stimulate it during this relative refractory period, this repolarization phase. Now, I don't know the exact number, but it needs to be stronger than 15 millivolts. So maybe it's 20, maybe it's 25, but as long as we're stimulating it enough to where it continues to hit threshold, it will generate an action potential as soon as it has completely reset. Now, why are these important? Why are we talking about these? As you can see, one, it prevents the action potential from going the wrong way, right? We want to be able to make sure that the all or none principle happens. So during this depolarization phase, there's nothing causing it to go halfway or stop or go backwards. It's all or nothing. If we hit that negative 55, we will go all the way to positive 30, no matter what. And it also keeps your neurons from frying, basically. It prevents them from just getting overworked. It forces them to rest. So it keeps them from just building on top of one another, where we have this too much electrical circuitry going on, and we can fry the system. So that is the refractory period. The relative happens during repolarization. So R for relative, R for repolarization. And the absolute, where nothing can go on, nothing else can exist, that's during the upward swing, what we call depolarization. And you can use this slide just as a summary page to kind of bring in all of the ideas and processes that we've talked about. So the neuron maintains the resting membrane potential at negative 70 by the sodium-potassium pump. Depolarization is initiated due to sodium flooding into the cell. Once that action potential starts, it moves like a wave across the axon, and then so does repolarization. And that refractory period, we have two types, right? We have the absolute, which I'm showing you in green right here, happens during depolarization. Nothing else can create another action potential. And then the relative refractory period here in purple happens during the repolarization phase. So this slide is a review of the synapse. We've already covered all of this in the muscle physiology chapter, but the only difference is we're just, instead of neuron talking to a muscle, we're just looking at a neuron talking to a neuron. So remember, a synapse is just the communication, a neuron talking to something else. And we have some structures here. So the synaptic knob is this knob-looking structure here. That's all vesicles with a neurotransmitter in there. So that would be the knob in purple. Once again, the cleft is the space. I'll draw the cleft in black. That's a space between the pre and the postsynaptic neuron. And then the plasma membrane of the postsynaptic cell, that's where those receptors are going to be for the acetylcholine. So that's just going to be this area in whatever color that is, blue, green, blue. So review, we've already talked about this. So just a review of terminology. Once again, a synapse is just something talking to something else. So in this case, a neuron talking to another neuron. And we have the designation of the presynaptic neuron that's delivering the neurotransmitter. And the one receiving the neurotransmitter would be the postsynaptic neuron. Now, there are two types of synapses, like I said. There are electrical synapses where we have a continuous membrane area. Some parts of the brain, this can occur. Also, you can see your heart and smooth muscle cells are connected, and basically they just have these tiny little junctions, these tiny little channels where ions just freely flow forward and backward. And therefore, you can generate action potentials that way without the need of acetylcholine or another chemical. But by far, the majority of the synapses in your body are neurochemical. They are with the help of some type of neurotransmitter, acetylcholine being the main one. So what exactly is a neurotransmitter? As we talked about before in the muscle physiology chapter, a neurotransmitter is just a chemical inside of a nervous tissue structure, mainly a neuron. And we can have different classifications of neurotransmitters, but the main way of describing them is, does it have an excitatory effect or an inhibitory effect? Does it excite or stimulate something, or does it do the opposite and shut it down? Now, what really is important here, though, is the thing receiving the neurotransmitter, the membrane, or really specifically the receptor receiving the neurotransmitter. Because as you can see in this bullet point that I just starred with the green star, the same molecule can be inhibitory in one scenario or situation, and it can be excitatory in another. So check it out. All throughout Module 3, we talked about how acetylcholine is so important for skeletal muscle contraction. You absolutely need it. You can have all the troponin, tropomyosin, sarcoplasmic reticulum, ATP in the world. If you don't have acetylcholine, that whole domino effect falls apart. You aren't contracting your muscles anytime soon. But take a look at what it does in the heart. That same chemical, no change, actually slows down the heart rate. And the reason why, and muscle contraction of the heart, but the reason why is because the receptors are different. The receptors in skeletal muscle are very different than the receptors in cardiac muscle, so they respond differently to the chemical. So the big difference is the mechanism of change in the postsynaptic cell, and that's due to what type of receptor we're dealing with. Now, what actually is a neurotransmitter? It's just amino acids. It's just the building blocks of a protein, right? The beginning stages of a protein. So you can have amino acids, amino acid derivatives. You can have small, large neurotransmitters, and basically, acetylcholine is a pretty small one for the most part. So everything on this slide is a review. We've talked about every single part of this with the neuromuscular junction. The only addition would be this last bullet point right here. What happens to the acetylcholine once it activates the postsynaptic membrane receptors? We do recycle it. We do reuse some of it. We basically break acetylcholine in half, and then it kind of just gets sucked back up into that presynaptic neuron, and we can store it or put it back together in the vesicles and reuse it for later. So it doesn't go to waste, which is a good thing. All right, and then we arrive to the last slide of the chapter, plasma membrane of the next cell. So this is just a zoomed up version showing you what's happening. And we already know how this works. Acetylcholine, or the neurotransmitter, binds to the proteins. So do me a favor and just look at this top picture real quick, because the bottom one is doing something different, which I'll explain. But the things in red, that's the neurotransmitter, or the acetylcholine as we've talked about. They bind to the protein, and that causes the protein to open, so then sodium can flood in. And if it's a muscle cell, it goes down those T tubules. If it's a neuron, basically it's going to start a new action potential. As long as we hit that threshold, right? As long as we have enough sodium coming in. Now, this bottom slide, same principle. We're going to talk more about this with the endocrine chapter next semester, but it doesn't always have to be directly correlated with that same protein receptor. So what you're looking at here is the neurotransmitter, same thing, the red things, bind to the protein receptor. And what that does is it causes a domino effect inside of the cell. And what happens is other protein activated can activate another protein, which causes chemicals to be released. And then chemicals can tell other proteins to open so ions can come through. And once again, we'll talk more about this next semester with the endocrine chapter, but a lot of our water-based, a lot of our amino acid-based hormones work this way. They kind of create this, what we call a second messenger effect. So ions and, well, the hormone itself can actually get inside of the cell. All right, everyone, that is it for nervous tissue and neurophysiology. Next chapter, we're going to be talking about the actual structures. We're going to go into great detail of the brain and all the brainstem structures and what they do, and the spinal cord. We'll even talk about some spinal nerves and cranial nerves as well. So I will see you next time. Take care.